Hello, good morning. I'm Zach Grinspan. I direct the pediatric epilepsy program at Weill Cornell Medicine, and the clicker works. These are my disclosures. I want to start with thank yous. What's that? I wanted to start with thank yous. Amber's presentation early, her fight like a mother, and what did you say? I'm going to be like the third monkey getting on the ark. That kind of drive, inspiration, energy has driven so much of the rare disease work that many of us are involved in, and has certainly looped me in and gotten people very engaged and excited. And I've said this before, but there's nothing quite like being on a research call, thinking about a study design, and a parent is on there, lighting the fire underneath your feet. It just keeps things moving and keeps it all real so that we can move this forward. The other thing is that the fundraising that happens at the foundation level gets this all started. So we got this trial started. We got the drug for free from the company, but the funding to get us going was through foundation support. And then Horizon, which is now Amgen, then gave us some additional funding to get this study up and to keep the study going. There we go. And then I also want to thank the two teams. The study I'm going to tell you about was done both at Cornell and at Colorado Children's, and there's a number of individuals that have worked tirelessly to make this happen. I'm going to tell you about a drug, 4-phenylbutyrate. This is a drug that is already FDA approved for something else. This was approved for urea cycle disorders. It lowers serum ammonia. It just so happens that it crosses the blood-brain barrier, which was very lucky for us, and it comes in two different flavors. There's sodium phenylbutyrate, which is approved first. This is super gross. It's very bitter and very salty. There are now taste-masked versions of it that some people in the SLC community have tried that aren't so bad as they were. But because of the poor taste, they came up with a second version, glycerol phenylbutyrate, and that's the molecule, Ravicti is the brand name, that we've used in this study. What happens is you've got a glycerol backbone, and then those phenylbutyrates pop off when you drink it. There have been a number of studies that have suggested that this molecule may work for developmental and epileptic encephalopathies for several different disorders. For example, STXBP1, which is involved in opening, in synaptic release of neurotransmitters. SLC6A1, that's this group, which is involved in the reuptake of GABA from the synapse, so it can be reused, recycled, repurposed. Across the hall, the GABA A receptor groups, there's, and Dr. Kang is here, who's done work both for SLC6A1 and GABA, and it may work for these disorders also. GABA is sort of the main listener for inhibition in the central nervous system. LGI1, there's a paper from Japan. This is a molecule that helps connect the presynapse and the postsynapse and helps organize synaptic activity. SLC6A8 is a creatine transporter that helps with energy metabolism in the cell, and may also have some work. May also have some effects at the synapse. SYNGAP, just next to us. SYNGAP is a postsynaptic protein that helps regulate synaptic plasticity, and then there's some emerging data. It may work for Dravet syndrome, STN1A, which is a fundamental sodium channel blocker that helps regulate overall cellular activity and excitability, particularly in GABAergic neurons. So what links all of these together? I put a question mark because it's not really clear. Best guess, they're all epilepsy and seizures. That's why I'm here as an epilepsy doctor. All of these impact synaptic function in one way or another, and all except for SLC6A8, which is X-linked, they're all heterozygous disorders, meaning you have one good copy and one bad copy. So how is phenylbutyrate supposed to help? High level, there are several different cellular mechanisms that have been proposed, and these cellular mechanisms have some functional impact. The list of mechanisms, oh, that came out weird. The list of mechanisms is broad, and every time I talk to a PhD scientist who works on this, they feel very passionate that it's one of these and not the other. But it is a big list. regardless of which one it is, it's going to have one of 4 impacts. Either your good copy, it makes that one work better. Either it's more stable, it sticks around longer, or it's enhanced and it works even better than it usually does. Maybe the mutant is rescuable. If you can just help the mutant get out of the endoplasmic reticulum or help it last a little longer, then you'll get more function from that salvageable mutant. Or maybe you've got toxic mutants, and maybe the toxic mutants aggregate or cause some other problem, and you can protect the cell against those negative effects. And finally, maybe it's something else. Other compensatory mechanisms, cellular biochemistry is extremely complicated, or maybe it's just another anti-seizure drug, which would be lovely. I mean, I don't want to say anything bad about that, but it may be that it works similarly to some of our anti-seizure medications. Anyway, we thought that the discoveries were important enough in the lab with a safe drug that the FDA has already signed off on, and so we opened a trial to try this in kids. The study design, it's an open label, multiple dose, single treatment group. That means that there was no control group, everybody got the drug, and we gave it to them every day, 3 times a day. Some kids have been taking it now for several years. Once we enrolled the patient, we would sort of follow them for 4 to 6 weeks, get a sense of what their baseline seizure frequency was, gather their data, understand their stories better. They would then come either to us in New York or to Children's Hospital Colorado for a 3-day, 2-night EEG admission. Early on, we waited 6 weeks, and then later on, we extended that to 12 to align closer with classic epilepsy trials. And then we would bring people back for another 2-day, 1-night admission, and then follow them a little bit just to make sure that they were stable. On occasion, people would then say, "Thank you, we don't want to be on this anymore." We would wean it off, and they would be done. The majority of children in the study have stayed on. We have an extended use phase now through December of 2025. I am so, like many people have been asking me for this for several years now, and so it is out there. This is published. Our early results, safety, tolerability, seizure, EEG outcomes for STXBP1 and SLC6A1 is now on this preprint server. So this is available to the general public. It is under review, fingers crossed, at a journal, and hopefully we'll get favorable reviews and see this published in the near future. Safety and tolerability, what a dream. So no new adverse events. When we first started this, you always kind of have the parade of horribles in your head, and you're like, well, what happens if we give this and they all go into status epilepticus? We designed the study for children to get the drug in the hospital for that reason, but serendipity favored us, and there were no new side effects. There are some side effects, and some of them are benign and some of them are less benign. There's a honey-like odor. Sometimes people think it smells like urine, so it's not so awesome, but it's tolerable. Some kids get sleepy. Some kids get appetite suppression. Metabolic acidosis has been the most challenging side effect to manage and has landed some kids in the hospital and had some kids decide that they don't want to be in the study anymore, their parents. As neurologists, as epilepsy doctors, acidosis is a side effect we're used to managing because we use ketogenic diet for many of our kids. So this is manageable, and we do have some kids where we're treating the acidosis with Bicitra, with baking soda, or other medications for that. Not a lot of missed doses either. Kids seem to take it. The way we published the tolerability is we said that many of the kids were continuing to take the goal dose or within a few percentage of the goal dose, suggesting that it's well tolerated. 5 children have withdrawn from the 48 that are now in the study, some for lack of efficacy and some for those side effects. So I have slides for 4 disorders. I'm going to spend time in SLC6A1 for y'all. In STXBP1, we had 6 responders. They had reduction in seizures. Many of them still have seizures, but we did show at 10 weeks that there was a nice reduction in seizures. 2 have a sort of a middle response that we couldn't really classify, and then there were 2 clear non-responders. All right, let me linger on this slide because this is SLC. So in the 10 that we treated, we're calling 7 responders. I want to hearken back to some of the things that were said before. So Dr. Varnett talked about how the kids, like, they have their eyes blink a little bit, and Scott and I talk about, Dr. Demarest and I talk about this quite a bit. Are those seizures? Are they stereotypies? What do we make of them? I'm not sure, but they got better. So what we did is we said that there were 5 children who clearly had seizures, and so we felt really good saying there was a reduction in those. There were 2 where we're like, I don't know what these are, but they went away, and so we're calling them spells. And then there were 3 who we ended up calling indeterminate. And the narratives here were interesting. There was one child who did not have seizures at the beginning, and then later, on Ravicti, developed eye-blinking spells. We dose-adjusted for the child's weight because he had grown, and then the seizures went away. There's a second child where it didn't seem to be much of a clinical response, but the dose that family is taking is about 1/3 of what we had recommended. And then finally, there was a child who withdrew initially for metabolic acidosis, and then the seizures came back about a year later and has re-enrolled and is doing well. Acidosis continues to be really challenging for that family, but we are managing it. One theme that has been really interesting with this group is that there seems to be something special about getting to the top recommended dose that the FDA has signed off on. We've had several kids, I think in the responder group, 5 of those 7, where we got seizure control with the Ravicti. They grew, and then the seizures came back, the eye blinking came back. So what we realized is that the children had grown, and so the mgs per kg per day had gone down, and inevitably what we've seen is that as we increase the dose again, those spells go away, which gives us confidence that we have an effective therapy, and also, as we work on access and more in the community start to use it, I have to tell you, those families were, it was often devastating that the seizures came back, and it was heavy for them. And so I want to spare some of the new families, as we were able to get this out to people, some of that heaviness, so that there is an expectation as children grow that they may come back, and it does seem to respond when we increase the dose. Things that are not on here are the development and the EEG outcomes. I have a slide on development I'll get to. With EEG, Dr. Searcy, this is fascinating. So there is a clear biomarker, right? And one thing that I think I've heard from you and from Dr. Goodspeed, there was some event where a kid was in the trial, and you called and you said, what happened to this kid? Because the EEG biomarker went away. And it was a Ravicti kid. It was a kid who had been in the study. So we've seen that clinically. When we reported in the paper, we were relying on the EEG reports that my colleagues had drafted clinically, and those trends don't seem to show up in our clinical reports, so we have work to do. But there does seem to be a lot of legs to this EEG story. All right, and then this is SYNGAP. We've given it to 9 children with SYNGAP, and we're seeing 6 responders in that group. Finally, GABA-A, we've given it to 5 children in GABA-A, and we have 2 responders. So I wanted to make a plea as my last couple slides. The natural history data is amazing, and we need more. And I'll show you why. And then the EEG biomarker data is so important, because if we have a way to track disease severity, it's going to make the trials much easier, and then clinical care when we actually get some of these therapies out to know whether they're working. Let me linger on these 2. So for natural history data, let's pretend that we have some developmental measure And you can imagine this space, chronological age on the x-axis, age equivalent on the y-axis. A neurotypical kid follows the diagonal. As they grow, their chronological age and their age equivalent grow. Dr. Miel is rolling her eyes at me because age equivalent has problems as a measure. But thanks for your patience with me. This is to make the point. If you have a child with a DEE like SLC 6A1, they're not going to be the same as that neurotypical child. So they're going to have a different trajectory. Maybe they plateau, maybe it's just a little slower, but it's important to know. So let's say we get a therapy and the children get better. They may not get back to normal, and we need that natural history data in order to understand the difference between a treated child and a not treated child. In an epilepsy trial, you get your outcomes in 3 months, but a developmental trial, it's at least a year and sometimes multiple years. And so the feasibility of doing a bunch of placebo-controlled trials with developmental outcomes is hard. But if there's really strong natural history data, you can sub that in as your sort of virtual control group and then make a strong argument about whether you think there's developmental outcome, whether there's developmental improvements. This is the Vineland data for the trial. The specific points are not being reproduced here, but the lines are sort of these fitted linear lines. Vineland is like the ABAS, a measure of adaptive skill, and you can look at receptive language, expressive language, and then gross and fine motor skills. What you'll notice with these trajectories is that in general, the children are delayed. We know that clinically, that's not news. Some of them, but not all of them, are getting better. And some of those curves are parallel or steeper than the neurotypical. What I don't know is whether this is from the drug or this is just what they would have done anyway. And so the importance of the natural history data is fundamental to our being able to fully interpret this to make sense of it. I would, these results are very exciting. It's a dream come true as a researcher to actually be able to make an impact on seizures, but I need to balance that by noting that some of the trajectories are flat. So there are children who are in the study who did not make developmental gains over the course of the study, who had already plateaued. And so I know that in the afternoon, several novel, super exciting therapies are being developed. I think when this story is told, Ravicti is going to be a first start, a first go, teaching us how to do trials with this disorder, and a bridge. But my anticipation is that some of these other therapies are going to get us all the way. And then finally, I always have to show these slides just to reemphasize again the importance of the EEG biomarker. The respondingest responder in the study was a 5-year-old who had spells like this, EEG and eye fluttering. This is one example. This is a compressed spectral array. Each of those little spicules is one of these brief seizures. This is 1 hour of data. This child was having dozens of these events. He started in Ravicti, got to a good dose, and when he came back 6 weeks later, the EEG had cleaned up. He had none of those spells, and I read this EEG as normal. Limitations, small sample size, open label study. Tonic seizures, like in the STXBP1 population, are very difficult to count because a lot of them happen at night, and so we know we're undercounting. As Dr. Demarest said, absent seizures and these eye blinking spells are difficult to count, and then we need the natural history data to make sense of it. My goal, just as I did for SLC and STX, we'll get the SYNGAP and GABA-A data out once we collect their 1-year outcomes over the next 12 months. We're having active conversations about making the natural history data more robust so that we can do the comparison of the trajectories of the kids in the study with the natural history and the EEG biomarker development. Literally the $1 million question is access. One of the roles that we can do from a regulatory standpoint in the US is we can get these compendia, there are 4 of them, that list on-label and off-label uses for various drugs. Many payers, Medicare, Medicaid, and commercial payers will look to these documents, which are not official, they're not federal, but they're very practical, for guidance on whether a drug is appropriate for a certain indication or not. And so we're going to work on these submissions. In Europe, it's very complex. But what I'm hearing is that the publication is helping, and then different jurisdictions have different requirements. So I heard from, I think in the UK, I'm looking at Lindsay because she's right here. In the UK, the repurposing office was like, send us what you got. And so they now have our preprint. In Germany, they want the peer-reviewed publication, so we're working on that as well. Thank you all, and I'll stop there.